Hello student. In previous lecture we have studied the protoplast culture. Today's lecture I am going to talk about the anther culture. Before starting to the anther culture, the first basic information about the organization of a typical flower. A typical flower consists of a four whorl of a floral part, namely calyx, corolla, androecium and gynoecium. These floral parts are divided into the two types. First, essential floral part and second, non-essential floral part. Among these, the calyx and corolla are non-essential floral part, which is also known as accessory floral part, and androecium and gynoecium are the essential floral part, which is also known as necessary floral part. Androecium. Androecium are the male sex organ of a flower. Stamen are the single unit of a androecium. The stamen are divided into the three types: long cylinder stalk-like structure called filament and upper bilobed structure called anther. And these anther and filament are connected to each other by connective tissue. The single lobe of a anther contains the two pollen sacs. In this pollen sac contain the diploid microspore mother cell. This microspore mother cell undergoes meiotic division to produce a four haploid tetrad of a pollen grain. In this picture shows the LS of a typical flower and shows whole flower part. Introduction about the anther culture. It is an artificial technique by which the developing anther at a precise and critical stage are excised aseptically from an unopened flower bud and are cultured on a nutrient medium. Here the microspore within cultured anther develop into a callus tissue or embryoids that gives rise to the haploid plantlet, either the process of organogenesis or embryogenesis. Rising of a plant from an anther is known as anther androgenesis. The first time Guha and Maheshwari, these two scientists, cultured the embryoids from an anther of a Datura inoxia in 1964. Now the type of anther culture. There are mainly two types of anther culture. The first, embryogenesis or direct androgenesis. Second, callusing or else indirect androgenesis. The first, embryogenesis. The immature anther are directly grown into embryo-like structure and these plantlets are regenerated from them are called embryogenesis. The callusing, the anther are introduced to produce a callus and then portion of callus introduced to initiate shoot and root and then produce a whole plantlet. Media for anther culture. There is no single media for anther culture. The composition of medium varies with the genotype of a species, age of a anther, then factor present in the anther wall. The MS, N6 and niche media are modified suitably to grow the anther culture. The composition of niche media in 1969. These are the components involved in the niche media and their composition or the quantity in milligram per liter. This table shows the component and quantity of a such a nutrient medium. Procedure: select or collect the unopened flower bud, then surface sterilization of this flower bud with 1080 and mercury chloride. After surface sterilization, wash with the distilled water. Anther are excised from flower bud and kept separately. Anther in the first meiotic division is selected for the anther culture with the help of acetocarmine paste incubated in the medium containing glutamine, L-serine and inositol. Then incubated the culture at 25 degrees Celsius for 15 days. Here anther grown into the embryoids. Embryoids are transferred to the rooting medium under 3000 lux illumination After 4 to 5 weeks, the embryos become a plantlet. These plantlets are acclimatized in a greenhouse. In this diagram shows the stages of anther culture. First select the unopened flower bud, then surface sterilization of such unopened flower bud with the help of mercury chloride, then wash with the distilled water, then dissected the stamen which is used or suitable for anther culture with the help of acetocarmine quash test. Then selected stamen used for anther culture and excise the anther from this stamen, and these anther are grown on a media. Then embryogenesis process is take place. After that the embryos is developed. These embryos transfer to the rooting medium. After that the grown plantlet. Then these plantlet are acclimatized in greenhouse for further growths. This is the colorful picture for anther culture. Here shows the two types of a anther culture. One first is embryos, that is, and direct introgenesis, and second one is calvation. Uses of a anther culture. Here used to produce the haploid plantlets. Utility of a anther and pollen culture for basic research cytogenetic study. Study of genetic recombination in higher plant. Study of mode of differentiation of a single cell to whole organism. Study of factor controlling the pollen embryogenesis of a higher plant. Formation of double haploid and are homozygous and fertile. Study for mutations, plant breeding, and crop improvement to obtain the secondary metabolites. Example: Hyoscyamus niger obtained by the anther culture having higher alkaloid content. Haploids are used in microbiology and genetic engineering, used to release new crop variety with superior trait. The main advantages of anther culture to produce the breeder pure homozygous genetic stock. Thank you for patience.